Okay, good afternoon, everybody. It's my pleasure to introduce today's speaker, Dr. Adam Roberts, who some of you may know joined us in February. He's here to drive our AMR and antimicrobial drug discovery programs going forward, and will eventually be housed in the new building. After his presentation, we'll find out who the real biologists and scientists are in the audience, because apparently this nucleotide sequence actually spells something, and so we'll be going round the desks later to see if anyone knows what it spells. If you translate it into an amino acid sequence, you've already been told, Peter. Adam's background is in applied biology. His first degree was from Coventry. He then moved down south, initially to do a PhD, and then 15 years at the Eastman Institute, looking at dental microbes. He learned the error of his ways, and we convinced him to come up north, and do things which are perhaps a bit more medically relevant than dental microbes. And he's going to talk to us today about all things AMR, antimicrobial resistance. It's in you. Adam.
Thank you very much, Steve. And thank you very much for coming on such a lovely day in the middle of half term. It's nice to see so many of you here. It's a pleasure to be here in Liverpool. And it's nice to know I'm going to be caged in the accelerator building shortly. Okay, so my title is slightly enigmatic, antimicrobial resistance. It's in you. I don't just mean resistance, I also mean possible solutions. And I'll cover both of those as we go through. Just some quick infographics for you first to kind of ground the problem. We've got here some images from the Jim O'Neill report. These infographics represent the number of people who are predicted to die per year of AMR, specifically AMR, in addition to everybody else who's dying sort of today. And you can see here, worryingly, that the majority of the deaths are predicted to be in the low to middle income countries. So the poorest in the world are going to feel the problem more than most. On your right here, you can see that actually the predicted number of deaths are set to overtake cancer, plus all of the others combined, by 2050 as well. So this comes with the caveat in the report that all bacteria will become resistant to all antibiotics, which obviously is probably not going to happen. However, it's a good starting point to stimulate discussion, because discussion is incredibly important when it comes to engaging the public, as we'll see. Two more infographics. An economist, Jim O'Neill, led this review. They wanted an economist because people really only listen if you talk in terms of cash. So you can see here the predicted problem in monetary terms is over 100 trillion US dollars. That's loss in gross domestic product by the year 2050. If we put that problem into context in this infographic, you can see that actually a £2 billion fund to stimulate drug discovery would be that pixel there. So it's actually affordable, but will we do anything about it? That's a different debate. So, in order to understand the problem of antimicrobial resistance properly, we really need to know all of the different things that drive it. This is one particular infographic I've got off the web. There are many of them. This one is found here. And you can see here that you've got different systems that we currently use. We've got food animals, we've got the environment, rivers and streams, aquaculture, drinking water. We've got clinical activities, food preparation. All of these things are interlinked, and we can't fix the problem of AMR just by concentrating on one particular block or network of these. In addition, this is not comprehensive. For example, this year there's been a couple of papers out looking at air pollution. Okay? Higher quantities of black carbon seem to give bacteria a little bit more tolerance to antibiotics because they affect the way that they grow within the lung. Tolerance to antibiotics can also facilitate the evolution of full-on resistance. Okay? So all of these things we don't yet fully understand, so we're still learning our way in order to try and tackle the problem. This is one of the major regions for our predicament as a species at the moment. We've had all these lovely antibiotics discovered and produced. In the last 30 years, we've arguably not really seen the introduction into the clinic of any new classes of antibiotic. So we really need to find some new ones. But actually, as a society, our reliance on antimicrobials goes a lot further back. So if we take a look, sort of pre-1900s, at what we do and how we've relied on antibiotics, we can get a context of the problem we're facing and how difficult it will be to change what we do, to change our behaviour. So in Greece and Serbia, for example, mouldy bread was traditionally used to treat wounds and infections. You can understand why, actually, in a modern day era. This is Penicillium notatum. It produces penicillin. You imagine rubbing that onto an open wound, and you've probably got some antimicrobial activity. Warm soil was used in Russia to cure infected wounds. Again, there's a lot of antibiotic producers in soils. It's the main source of antibiotics that we have today. Babylonian doctors healed the eyes using a mixture of frog bile and sour milk. Sour milk probably contains Lactococcus lactis, which produces many antimicrobials, and also bile salts are slightly antimicrobial in nature as well. So all these legends have a basis in fact. And also, to keep milk from turning sour, Russians put a frog in the bucket, which sounds remarkable. Until you actually read this paper, and based on this, they had a look at the secreted peptidome of the frog, and they could isolate brand new bradykinin peptides from it, which have antimicrobial capabilities. But actually, even before that, we were relying on antimicrobials. So as civilizations emerged and towns grew into cities, one of the main problems we faced was getting rid of our sewage. The water was disgusting. If you drank it, you inevitably got ill. If you continued to drink it, you probably died. So there's two ways that humans came up to solve this. They used tea in the east and beer in the west. And it was only by drinking tea and what actually became known as breakfast beer. It's not like a Stella or anything. It's not a normal fighting day. It was a very weak alcoholic beer, but it was cleaner than the water because of the antimicrobial properties. I like this one. So the mythology surrounding the wishing well, I believe, started when Odin, the Norse god, took an eye and threw it down a well so he could see the future. It was a trade-off, as it would be. But obviously that became embedded into myth by people offering silver coins. And it's the silver and the copper in the coins that actually gave a slight antimicrobial effect to the water in the well. So that became a lucky well. People who drank from that well were kind of thought of as better, so put another sacrifice in the well. Until it got toxic, it worked probably quite well. So you can see that our societal reliance on antibiotics is deep-rooted in our society, if you like. We've gone from a risk-averse society to an antibiotic-reliant society, and that's something that we may need to change. Right, on to my previous work. I want to talk about my previous work because I'm not going to mention everything I'm going to do here. That's for a different talk. So as Steve said, I worked at a dental institute for 15 years, and in order to keep your managers happy at a dental institute, you have to work a little bit on dental organisms. Now, dental organisms, dental bacteria, are actually quite interesting. They form a massive reservoir of antimicrobial resistance genes. So for somebody interested in AMR, it's a really easy sample set to get. In addition, they're an incredibly adaptable microbiome. You can do a lot of things to the mouth. If you imagine what your mouth has to go through every day, it's extraordinary that the microbiome relatively remains unchanged. Extremes in temperature, heat, carbon sources, etc. Even when you treat them with antibiotics, it very quickly shifts to its normal state. It's very adaptable. And I've just put, this is kind of my swan song from the dental world. We've worked out what actually drives your microbiome, and it's who you live with. It's your family rather than your host genetics. So this is currently under review at the moment. So I work with dental bugs from the mouth. It's a very easy site to access, and they're common. We've all got one. So it's very easy to find saliva. We usually find it from the international students that come into the department. And it gives us a great thing to work on. Now, there's many different environments within the mouth. and we cannot grow all of the bacteria that live in there. So what we have to do is use metagenomics. We bypass the cultivable step. I've got many projects looking at cultivable bacteria, but I'm not going to discuss those today. I'm going to talk about the metagenomic samples. So essentially what we do is take some saliva, we break it up, and we put it into a replicon, usually a back vector. This enables us to clone very large pieces of DNA. And then this ligation reaction we can transform into E. coli, okay, so a surrogate host, and then plate out, and we can pick all of these in, I say we. The PhD students can pick all of these individual colonies, and they can put them in microtiter plates. Many microtiter plates. Can you see those microtiter plates at the back? They're very ghostly microtiter plates. So the library that I've brought here actually contains about 30,000 clones, all in these microtiter plates. And they're all frozen down in the freezer. So if anybody's got an assay, come and see me afterwards. We've probably got the DNA. What we can do with these libraries is to screen it on antibiotics to see if there's a background of resistance to commonly used antibiotics in the oral cavities of healthy humans. The first one I'm going to describe is tetracycline resistance. So we simply screen the library out onto tetracycline-containing plates. Tetracycline is a very widely used antibiotic. The earth is effectively covered in a thin film of tetracycline, so resistance is quite common because there's a good selective pressure. One of the clones we found had a 7.5 kb insert, which contained 5 genes. These 2 genes looked of interest. They encode, or predicted to encode, an ABC transporter, which formed together to make a pump. If you imagine this is the cell membrane, okay, bilipid cell membrane, these are the 2 proteins which we predict to be encoded by those 2 genes. They span the membrane and they receive ATP, there's a dephosphorylation reaction, and then there's a small conformational change here and the substrate goes through. So we envisaged that that was how tetracycline resistance could be happening in this particular clone. This Walker A motif, which binds the ATP, this small section of amino acids, is critically important in binding that ATP. So we predicted that if we delete that, which was here and here, actually we should get loss of function. So we made an in-frame mutation to delete the Walker A motif, but not interrupt the not interrupt the transcription because they're slightly overlapping genes and there's no cognate promoter here. So we made those mutations, checked the minimum inhibitory concentrations, and you can see in here and here it reduced from the wild type. So this is the E. coli strain just containing the empty vector. This is the E. coli strain containing the 2 genes that we found that we predict to give rise to tetracycline resistance. This is mutant 1, mutant 2, complement 1, and complement 2. And you can see here that we've got resistance, loss of resistance, and then gain of resistance upon complementation. So a very classical genetic experiment. We also tried it for minocycline, which is a related analog of tetracycline, very similar in structure, and there was no resistance at all. And then my PhD student, Liam, had a crazy idea to try it on tigecycline, which is a semi-synthetic tetracycline with a bulky side chain on. And this bulky side chain was put on by chemists actually to stop it being excluded by efflux pumps, which is exactly what we found. However, when we looked, we found that actually resistance is conferred. So this was the first efflux pump conferring resistance to tigecycline in E. coli. Now you may say, so what? But actually, if you consider that E. coli now is becoming resistant to pretty much everything we can throw at it. So this is an E. coli which contains the MCR1 gene, conferring colistin resistance. It also contains the NDM gene, conferring metallo-beta-lactamase resistance. And the only thing you can kill it with is tigecycline. And we've got tigecycline resistance genes in our mouth. So you can see it's not going to take long if there's an increase in the use of tigecycline for those genes to be selected for and for the resistance to proliferate. So that was all quite worrying. Next, I want to describe not antibiotics this time, but antiseptic resistance. So cetyl trimethyl ammonium bromide, also known as CTAB, is a very common disinfectant. It's available in a lot of cosmetics. in things that you find in the kitchen, hand washes, etc., etc. So again, we've basically coated the earth, at least in the Western world, with this antiseptic. Antiseptic and antibiotic resistance can quite often congregate on the same mobile genetic elements. So you select for one and you select for the other. This microtiter plate represents the culmination of 3 months' work of one of my MSc students who screened the 30,000 E. coli clones of the library I previously mentioned, and that was his result. Can you see that little shadow there? That's what I said. No, I can't. So we plotted it on a graph. So there's 95 data points down the bottom here, and that's the one that he convinced me was resistant to CTAB. He isolated the plasmid. He showed that it contained a reasonably large insert, too large to sequence in the 2 weeks that he had left. So he carried out transposon mutagenesis, where you insert a transposon within the backbone and insert of your plasmid and then look for loss of function. And he got 8 out of 9 transposon insertions that became susceptible to CTAB in this gene here. One here, but I think that's a polar effect. These regions of DNA were then cloned, and the MICs determined, and he was absolutely right. This gene on its own was sufficient to give a doubling in the MIC. Now this is called an epimerase. So what does an epimerase do? It reversibly catalyzes the formation of UDP N-acetyl galactosomine from this one. And it toggles between the two. Okay, so you get a ratio of this versus this while you're building your cell wall. So that means any changes may affect the cell wall. So we immediately thought, actually, let's investigate the cell wall. So the first thing we did was strip the cell wall off, put it through a mass spec, and we found actually, compared to the E. coli with just the plasmid, the E. coli with the plasmid plus that epimerase gene had an interesting spike, but it wasn't actually reproducible. We kept getting different spikes, and we think that might be because of the conditions and a very finely tuned dynamic that was going on in the cell. So we decided to look at the charge of the cell instead, and we can see here, compared to the plasmid containing E. coli only, the charge is slightly more positive. This was in a cytochrome C assay. So this represents the first epimerase-based resistance mechanism to an antiseptic. And what we think is that the epimerase is making the cell more positively charged, and that is repelling the positively charged CTAB and CPC molecule. It's quite a novel global cellular response, leading to tolerance of these antiseptics. And then finally, another antiseptic is triclosan. So triclosan is in pretty much all hand washes. It's recently been banned by the FDA because it's quite toxic, and also it's promoting resistance. We wondered if we could find triclosan-resistant genes in the oral cavity. We found that actually, if we clone this, it's called a FAB-I. It's an ENO-A-SAR carrier protein. Again, it's involved in the production of fatty acid in a bacterial cell wall. If we clone this in our library, we got an increased tolerance to triclosan. We got multiple clones, and we got multiple species of insert, all containing the FAB-I genes. So every time we got a FAB-I gene cloned in our library, we got triclosan resistance, which was quite interesting as well. So it just goes to show that actually, if we've got a normal metabolic gene from a bacteria that's not normally involved in antimicrobial resistance, and we put that in a heterologous host, in this case E. coli, we can actually get a resistance phenotype. But that's all well and good. We did it in the lab. It's a library, so we made it. Is it all an artifact? Does it occur in nature? Well, yes, it does. So this is a paper from Marco Oggioni in Leicester, one of my collaborators, and he showed quite nicely that actually the FAB-I genes have made their way onto transposons and plasmids. Okay? And this is important because these genes, pretty much any gene, can find their way onto a mobile genetic element. And once they do that, their potential for transfer is greatly increased. So, one of the things we wanted to do as well was to see if we can identify mobile genetic elements associated with antimicrobial resistance genes within the oral cavity of healthy humans, none of which have had antibiotics in the previous 3 or 4 months. So I'll just give you a little bit of background about mobile genetic elements. They come in all shapes and sizes. Actually, that's not true. They are either circular or linear. They come in two shapes and sizes. You've got plasmids, which are self-replicating molecules. They exist in the cytoplasm of the cell, separate from the chromosome, although they can insert in and out very rarely. They're quite often associated with antimicrobial resistance. So you can see here on this one, I found this one from a streptococcus strain in a mouth, and it confers tetracycline resistance with this gene here, TET-S. There are also insertion sequences, which are very small elements, and these encode usually a single protein, which is responsible for their own excision and insertion. So these are very parasitic pieces of DNA. However, they can come together and they can flank other pieces of DNA and form what is known as a composite transposon. And then probably more clinically important, we've also got conjugative transposons, and these do everything that all of the other elements do. They're able to insert, excise, move around the genome, but they can also conjugate from one cell to another. So when these pick up a lot of antimicrobial resistance genes, you can see that you can get wholesale transfer between different genera, not just species. So we were quite interested in the composite transposons. Okay? It's a type of transposon consisting of two insertion sequences flanking a DNA segment. Evolution usually selects for this DNA segment which contains antimicrobial resistance genes or other genes of adaptive nature for the host. One of the things that it does is excise, and when it does, it can form this circular molecule which is called a translocatable unit. And this is quite an interesting one because it's a circular molecule, it's very small, but it hasn't got any replicable function. It can't replicate on its own, but it can homologously recombine with another copy of an insertion sequence somewhere else in the genome. Okay, so it's quite a novel way of transfer. So we were interested to see if we could find these and these in the oral metagenome. We decided to concentrate on composite transposons flanked by IS1216. It's very common, and it's usually been found to be associated with antimicrobial resistance genes. We really wanted to maximize our chances of success here. Additionally, IS1216-based composite transposons have been found before to be excised and form TUs. So we thought this would be a good place to start. So the PhD student designed primers after aligning all of the IS1216 elements known to humankind. He came up with some primers to amplify the internal region of any composite transposons. Multiple PCR products were obtained, and these were all cloned into an amplicon library and sequenced until saturation. In order to make sure we were working on a correct amplicon and not just something which was an artifact, the primers were designed slightly in from the ends of the element so we could use this as a signature. So we knew actually this conserved region, if it's there, it's a proper composite transposon. Some of the structures that we found were correct, IS1216 elements flanking it, and then genes in the middle. Now this one's quite interesting. This is QRG, which encodes for a quaternary ammonium resistance protein. So this confers resistance to CTAB and other antiseptics. So this is present. This mobile element with antiseptic resistance genes is present within our oral cavity. Next one is USP, was contained on a similar kind of element. And this encodes a universal stress protein, and this function of this is not actually known, but it's over-transcribed during many different stresses that the bacteria encounter apart from temperature. So while we don't know what it does, we can infer that it's likely to have a role in stress adaptation. So that worked with the IS1216. We thought we would try it with all of these as well. So different primers were designed based on the fact that all of these are found in Streptococcus, which is the most common species within the mouth. These are members of the IS6 family, which is probably the most common insertion sequence family. And these are often found on transposons or plasmids and are therefore linked with AMR genes. What we found was just another single one from IS257, which incorporated a kanamycin resistance gene here. So we've shown quite nicely that we've got composite transposons circulating within the mouth of healthy humans, and these composite transposons have antiseptic and antibiotic resistance genes within them, which is nice. Now the second part was to see if we could detect these circular forms, to see if they're actively excising, because remember this can't replicate on its own, so the only way we're going to detect this is if it's actively excising at the moment that we isolate our metagenomic DNA. Primers were designed from the individual segments that we found reading out, so we should, if this exists, amplify an IS1216 element. Those PCRs were carried out, and these two showed us that actually we could isolate the predicted circular form. So we've also got TUs circulated within a healthy human mouth. So this just shows that there's quite a massive background of antimicrobial resistance genes on transposable elements circulating within our saliva, if you like. And this also means that they're capable of mobility and selection if they can make their ways into different hosts. And the obvious example from our research is if there's a move to tigecycline resistance following the spread of colistin resistance. Okay, so that leads me on to an announcement at this part of the talk. A lot of these mobile elements, in fact most of them, contain antimicrobial resistance genes. And one of the things I've done since 2006 is to run a transposon registry. Before this, it was chaos. If you found a new transposon and you wanted to name it, usually you named it after your own surname or group or institute, and it was a complete mess. So in 2006, I instigated a new transposon registry, which was based on a previous one run by Esther Lederberg out of Stanford, but was left when she retired. And that systematically gave people who requested them TN numbers. So some of the elements that I've referred to here are TN6087, TN916. This transposon registry, which is now hosted at the LSTM, and I think is launched now, or at least within this hour, so it will be live at the end of the talk. We'll aim to do that again. Is that a transposon registry? Excellent, it's live. So visit this, tweet it, and what it will basically do as it expands is to form a repository for all known transposons associated with AMR genes across the planet. So it'll be a one-stop shop for all of the information that you need. So that'll be live now. Right, let's go on to the solutions. I've talked a little bit about the problems, about the fact that we all contain antimicrobial resistance genes. It is in you. What are the problems? Sorry, what are the solutions? So this is a 10-point plan which came out of the Jim O'Neill review. And it's quite nice actually. When I came here, most of these, probably apart from that one, are actually being done to quite a degree of depth. So it's really nice to see. Obviously, this is one of the ones that we really need as a society. We need new drugs. There's no escaping it. We can have adjunctive therapy, we can have phage therapy for certain things, but what we desperately need is new drugs. And for that, we need new funding. We need new funding because people are not looking for new drugs because of this. So this is the predicted profit return if you're going to make a new drug. This is almost $1 billion. You can see how much you spend, and then only on patent sales here, after maybe year 12 to 15, do you start to recoup your money. And you've probably got about 2 or 3 years before it becomes off patent for you to make a profit. So that's just a nonsensical business model, really. And there's a lot of things which are being suggested to try and fix that. But while that's being fixed, it's nice to see that actually better funded early stage research was called for within the review. And this has been echoed many times recently. And I just want to highlight this. This is CARB-X. Okay, so this is the Consortium for Antimicrobial Resistance Bacteria. And they just announced £48 million worth of funding for, I think, 12 early stage projects. So this is fantastic. It will work as a stepping stone to get your innovative ideas through to early stage trials. And this was only possible because of a political impetus globally, which has occurred over the last few years. And I just want to kind of go in how you get this political impetus going. You start off with experts, and then you have the Chief Medical Officer, Dame Sally Davis, talking, convincing Dame Sally Davis of the importance of AMR. She then convinces the then Prime Minister David Cameron of the importance and the fact that we should commission a review. He commissions Jim O'Neill to make the review, who then discusses with Sally and the experts what should go in the review, and then publishes the review. And that took a while, and that was published at just the right time to complement the World Health Organization Global Action Plan and a lot of public engagement activities, again catalysed by Dame Sally Davis, including the Longitude Prize, which I'm an advisor to. So this is a massive prize, £8 million prize, looking for rapid diagnostics. And this was put to public vote. So the Longitude Prize is supposed to solve one of the greatest issues of our time. There were 6 categories. Okay, antibiotics was championed by Dame Sally Davis, and there was a massive push to get the public to vote for that, and it worked, and it won. So there's sustained public engagement with this topic through the Longitude Prize, which is excellent. And it all went to help and push David Cameron into getting this on the political agenda. Following the publication of the O'Neill Review, it leveraged inclusion at the G7 in 2016, and it formed part of the write-up. You can see here, I think it's point 3, yeah, AMR. So that was the first time it went on to the G7. Because of that, it also then went to the G20 in the same year. You can see Teresa there. Yeah. And this made it to point 46, which is just above the thank yous. But that was just enough. That was enough to get it into the UN high-level meeting, where all of the countries within the UN signed and ratified an agreement that they would do something about it. They would put together an action plan. And it's that action plan, actually, that the states signed up to and then made CARB-X as a response to that. So it has worked. Unfortunately, what hasn't worked is the public engagement aspect of it. So it's on the political agenda now. There is sustained funding. At the B20 meeting last week, again, it was reiterated that AMR is an important area and needs to be tackled. But the public perceptions still baffle me a little bit. This is a tweet that I took off Twitter this morning. It was very light this morning, I couldn't sleep. And you can see here that the BBC4 are interviewing a woman who is suffering from antimicrobial resistance because she was prescribed too many antibiotics as a child. So you can see how wrong that view is. And then also Craig, who works over the road, he said this morning, sure, the BBC said that the new vancomycin, so there's a new vancomycin been described, vancomycin 3.0. It's got some different chemical moieties on it, and it works a lot better against gram-positives. And that was said to be, it won't be resistant to the bacteria. So all of these things are mixed up. They're all wrong. Until we get that public perception correct, until we educate the public, there's no way that we can tackle it from a public-led level. So I think this opens up an opportunity. I'm quite passionate about public engagement activities. I'm paid for essentially by the taxpayer, and therefore I think I have an ethical and moral obligation to engage the public in the research that I do. But I've often sort of been troubled with how to do that properly, especially around the topic of AMR, because it's a bit doom and gloom, isn't it? You say 10 million people are going to die by AMR, but actually we're all going to die anyway, and there's lots of other problems competing for the space in people's minds. Trump, global warming, Covfefe, Brexit. Okay, so it just doesn't really work. So I was thinking about how to re-energise this kind of public engagement activity, but also link it with my research. Now, there's a few projects in drug discovery which I'd heard about and I really liked. One is Ancient Biotics from the University of Nottingham, where they look at old recipe books in their spare time. and then try and replicate those in the lab, and they've shown that they work. You know, if you take eye of newt and some garlic, then it kills MRSA, and that's really good because it captures the public imagination. Matt Hutchinson over in Norwich, he uses leaf cutter ants and their symbiotic relationship with the fungi and the symbiotic relationship with the fungi and the bacteria on their cuticles as well, to look for new antibiotics, to maintain that symbiotic relationship. And that's worked really well, actually. He got a paper out earlier this year on a new species of bacteria making a new chemical. And you can see here one of the authors of the paper said, our findings highlight the importance of searching as yet underexplored environments. And I really agree with this. I think there's so many places on the earth that we haven't looked at, that there's a real opportunity here. And I don't just mean inside million-year-old cut-off cave systems and the bottom of the ocean, although obviously if I had the funds I would go there. What I mean is everywhere, day to day. Oh, and I must mention this one as well. This is Antibiotics on Earth from the Microbiology Society. So this is basically looking at soil and trying to engage the public with that. As I said before, soil is the place where most of the antibiotics that we use today have been identified from. So I was thinking, you know, hope changes everything. So why don't we do an antibiotic discovery project where the public can decide where to look? They can make the decision and they can then have ownership, if you like, of their research. They'll be more likely to engage. And then came the problem of who's going to pay for it. UCL wouldn't, I know Steve wouldn't. The research councils wouldn't, because actually that's kind of unethical. It's a bit of a stamp collecting duty and a bit of a survey, and it's not really guaranteed to succeed. But I thought actually the public would get behind it so much that they would pay as well. And they did, which is nice. So we launched a project called Swab and Send. It's got a website, it's a Facebook page, it's open. You don't have to be a Facebooker to look at it, but I advise you to go on. The whole of the history of the project is on the website. And what we do is if somebody pledges a very small amount of money, I think £30 for 6 swabs at the moment, or £30 for 5, I'm not sure which, we send them the swabs and we send them an information sheet. And we've had samples from all over the country and also from other countries, Icelandic glaciers, the pyramids, forests in Canada recently, and inquiries from all over the world. So people really want to take part. And on that form as well, there's a space for them, well, primarily for the kids who take part to draw diagrams and really engage with it to tell us what they've found. Actually, what we noticed was that the adults like to take part. So we've got here, for example, a map of Guernsey, which is almost identical to the Ordnance Survey one. We've got the largest piano I have ever seen, which has been swabbed recently. And some people really get behind it and actually send in photographs of where they swab. And this person stalked me on Twitter a little bit. But they're really into it. But the best one, I have to say, which really makes me believe that we have future scientists in the UK, is from a 7-year-old boy. I can't remember which school he was from, but he sampled a dark corner. I just think that's absolutely brilliant. I've also had the privilege of being invited to and swabbing various places of note, at least for a microbiologist. So this is one of the original plates that Alexander Fleming had. I asked about this crack here in the agar, and apparently Alexander Fleming was not only quite an astute scientist, but he was also a bit of an artist. So he introduced that with a scalpel to make it look like a flower, which is quite cool. I like that. Anyway, I swabbed that, and I got something that could kill MRSA from it, which was nice. From the outside, I hasten to add, not from inside. That's it's sealed. I've also swabbed boats, such as the SS Great Britain, with school kids. Daleks have been swabbed by Adam Rutherford at the BBC. And bees. So this won a photography competition, which was great to promote the project. This is the kind of thing we get. We take the swab, we plate it out on a non-selective agar plate, which is nice and rich and nutritious. This is from the floor of Liverpool Street Station concourse, just before my PhD students were arrested. That's another story. And what we can see quite nicely, this one's from honey actually, and you can see here there's a zone of inhibition. So whatever this is inhibiting this mold here. So you can get instant results, if you like, and then we can follow that up. But this is kind of a work through on what we do. So this is from my daughter's school in Worcestershire, and just keep your eye on sample 29. This was from a computer workbench. So all of the samples are plated out to purity, and then they're all picked into a microtiter plate. Okay, so these numbers just refer to the samples. 29, we've got these two different ones here. They're plated out using a hedgehog, not a real one, a 96 well pin replicator, onto large agar plates and then replicated onto agar plates which have had the indicator strains streaked along the background, and you can see here the zones of inhibition. So these isolates here are producing something which is killing this background strain here. And you can see 29 has got a little zone of inhibition as well. We use multiple indicator strains. This is the weakest but most photogenic, which is why we use it, and that's Micrococcus luteus. It's an environmental bug. The bacteria that could kill that, it's not really indicative of whether we want to use it for medicine, but what is, we have MDR E. coli, so multi-drug resistant E. coli as an indicator strain, MRSA and Candida albicans. And this just shows the Candida albicans plate. So this is an isolate from number 29, and that's the best zone of inhibition that we've got against Candida albicans in thousands and thousands of isolates which have been tested. So we're quite interested in this, and this will be plated out next week for continued research. We only take these isolates forward if they kill the Micrococcus luteus, the MDR E. coli, and or MRSA and Candida albicans. And we've narrowed it down to about 20 at the moment, which we're looking at, all fully genome sequenced, trying to find out exactly what they're producing. This is just an idea of what has been swabbed. So cats, even though I say don't swab animals or yourself, everybody does. So this, you know, the solution may be in you if you take part and you decide not to obey the rules. So the cats are always quite useful. There's quite a lot being produced. The chief executive of the BBSRC had a go as well and had quite a dirty desk. The BBC seemed to love it. There's a whole plethora of different areas which we can sample. One of these from a child's trampoline is one of the ones that can kill all of the strains that we've tested it against. So we are looking at that as well. And these are the first results from the LSTM last week. Okay, these are just a small section of the isolates that we've got, and this is against the Micrococcus luteus. And you can see here where the samples were isolated from, obviously river otter poop. But this salmon carcass isolates, these two here, they could kill they could kill Candida albicans and MRSA as well as the Micrococcus. So it just shows, it doesn't matter really where you look, you might be lucky. So that's what we're aiming to do. And finally, I just want to go back to the website. You probably can't see this. This is just a post showing that we can get anti-Candida activity, and there were 349 people engaged with this post early on. And then a couple of other posts. This one on New Year's Day in 2016 got 2.1 million hits. And then this one, which was a bit of work I did with beards last year, got over 6 million views. So we've engaged over 8 million individuals with this project already in the space of the last 12 months, which is great news. So just to conclude this section, swab and send. We've got hundreds of swabs being sent back to us. We've got thousands of isolates, all been screened against multiple indicator strains. These are all now in the freezer here, and they will be screened against other organisms as well, bacterial and otherwise. We've got multiple leads which inhibit all of these, and we've managed to reach millions of individuals and engage them about AMR in a positive way, which I think is quite important. Okay, so to conclude, AMR is a multifactorial problem requiring multiple linked solutions. There is not one answer. We've got to work across the board, kind of a One Health approach, which is what we're doing here. We need to understand what is circulating within us and the environment, agricultural systems, etc. Because a small change, for example, in therapy, may lead to a drastic difference in selective pressure. New drugs are essential. Funding for new drugs shows signs of increasing as political impetus gathers, although I think this is at risk now with current events in the world, but we shall see. And I believe that there is space for academic drug discovery projects at the moment, and we can de-risk those by providing fundamental science and public engagement returns. And also we've got to remember, if we don't look, then we're never going to find it. And with that, I will just thank the individuals whose work I presented today: Liam, Supertep, Marco, Lee Chin and Kimmy, and then the funding agencies who made it all possible. And I'll be happy to take any questions. Thank you.
Any questions for Adam? Anyone with questions, if they could also say who they are when they have the microphone. So, David Hong.
Hi, Adam.
Hi.
The SWOT project seems to be very, very interesting, but I would like to hear a bit more about how you plan to take that forward if you found some swab sample which you think it's worth to taking forward.
Yep, it's a good question. So the first thing we do is to sequence the genome. Okay, it's relatively quick and inexpensive these days, and then we can run that through various pipelines such as anti-smash to see if there's identifiable biosynthetic gene clusters present in the genome. If there is, and it looks like we can predict exactly what it's producing, we will probably leave it at that stage because we're interested in new biology and new chemistry. If it makes it through that, then what we'll do is to look at cell-free supernatants and then follow the activity with fractionation, mass spec, to try and work out exactly what the molecule is here. So we'll always follow the activity until we know what it is, and then we'll leave it, or if we can't, we'll discover exactly what it is. It should be new.
Adam, I thoroughly enjoyed that. At the start, you said there was this big network of impact or inputs into this
Yep.
development. I've heard arguments swing from which is the most important driver of this. In your view, is it people not finishing the course of antibiotics, or is it use as food growth promoters in agriculture? If there was one thing you were able to, with a magic wand, get rid of, what would be your priority?
Use in agriculture and aquaculture. It's not just growing pigs in China. The use in Africa, the use in America is incredibly rife. The use in fruit farming is incredibly high. Fruit trees are sprayed full of antibiotics. I saw a couple of slides from a talk in Bristol a couple of weeks ago where farmers were injecting trees with antibiotics. Like 100 mils of them, 3 syringes going into the base of the tree to promote the growth. And it's just absolutely crazy.
And is anybody trying to do anything to prevent that or educate farmers in different usage?
Well, I think they are. I mean, so in the States, for example, there's a political impetus now to try and get rid of it, but the farmers' unions and the producers of antibiotics are very powerful in their lobbying. So there's kind of a stalemate at the moment. But there seems to be a trend where end users, so the public who buy the products, are moving away from intensively farmed products which are grown under antibiotic prophylaxis, if you like, or growth promotion. So it seems that there's a new driver which is public pushed sort of perceptions, and actually we want healthier products. We don't want to use eggs which have been farmed from chickens that are pumped full of antibiotics. So it seems to be happening. In low to middle income countries, it's a lot more difficult. With colistin resistance emergence from China, The Chinese government, to be fair, very rapidly tried to de-risk the situation by saying we're not going to use colistin anymore, and they used thousands of tons of it. So they're moving away from the use of any antibiotics in agriculture, which are used primarily for human medicine. So there does seem to be a general shift away. But as new industries and societies emerge, I think there is a very big danger that generic antibiotics will be used to underpin most of agriculture, at least on the initial stages.
I think that's really interesting. But what we have here a lot is a lot of parasites. We've got a lot of venom proteins. We've got a lot of different proteins from different sources. But one of the things I wanted to just throw out as a stupid idea is helminths are associated with a hygiene hypothesis, which has been thought to be autoimmune related. That's probably right-ish. Helminths then go through life cycle stages that will have the opportunity to pick up all sorts of materials in their free-living life cycle stage and then deliver that directly into the humans, whether it's in through the gut or through the blood. May that be something that's worth looking at in terms of either delivery of bacterial disease and what you're looking at?
Possibly. I suppose the sample sets exist, don't they? Because we've got sort of humans without helminth, populations of humans without helminths these days, compared to those in which it's still endemic. So we could look at those.
Follow on from that, it would be really interesting to look at the gut of gut worms, see if they share any of the microbiome that their host is, what they surround themselves in and feed on, or whether or not they have their own unique microbiome and what the interaction is there.
That would be, yeah, I agree.
You could find a bucket full of asterisk, I'm sure.
Yeah, I've got some. We've got one this afternoon. After lunch.
Yeah.
I get the impression that you've got more than enough to be dealing with at the moment, actually. But a comment on the observation that when the scientific community was using the strategy that you have highlighted there at the end, which seems to be relatively straightforward but was very low throughput, we managed to generate many classes of antibacterial compounds in the 30s, 40s and 50s. And when all the clever genomicists came along and gave us targeted drug discovery strategies, and we targeted hundreds of millions of compounds against essential genes, we came up with 0.
I agree. It doesn't seem to have worked very well, does it?
Any further enlightenment on why one simple low throughput mechanism worked and the sophisticated high throughput system failed?
So with the low-tech system that I'm using here and has been used before, basically all of the evolution of this biological warfare has been done before. So these natural products, they've been honed over millennia, millions and millions of years to be absolutely right for purpose. And that is to give the producers a competitive advantage within their particular niche to kill or prevent the growth of other bacteria. So they have an advantage and they can thrive. That's how evolution works. What we did when we came along and used synthetic libraries, etc., was really take a massive chance. And it didn't really work that well at all. And when it did, there were a few instances where it did, and we got some synthetics. Resistance actually existed already. It was just a different function at the time. But when these new molecules came along, resistance emerged really quick. So I think with the natural products within soils, fridges, gardens, trampolines, wherever we may look, there's still a huge repository of novel chemistry to find. And that's what I'm hoping that we'll capitalise on.
Last question.
Could I carry on the same sort of theme, really? Because it occurs to me that if you're a pharmaceutical company about to spend $1 billion on the development of a new molecule, it makes you quite risk averse. And the number of hoops that a molecule needs to jump through in order to satisfy the public has increased quite remarkably over the course of the last 50 years. so that, if you like, the killing rules for any particular project are now extremely strict, and the company will kill the molecule quite readily if it doesn't obey certain principles. I wonder, in your engagement with the public, who of course elect the politicians, whether one aspect ought to be greater awareness of realities of risk. That is to say, even this magic vancomycin, which makes it onto the radio, is actually bloody toxic, and one has to take the rough with the smooth.
I agree. I think it's vitally important that there's a full understanding of that. But I think to do that, we have to battle against the media, like the BBC, who, it's a buzzword. We're all saved. Woo. Actually, it's never been tested in vivo, that new vancomycin. And it's just for gram-positives, which is not really what we need, because we've got bucket loads of those anti-gram-positive agents.
The textbook I read as a student had a little graph which showed the lifespan of a new drug, which was best thing since sliced bread, and then an adverse event, wouldn't give it to a dog,
Yep.
And then useful under some circumstances.
Yep, I agree. I mean, you can't almost pass a clinical trial, can you? You fail them, or you get passed. It's very cutthroat. But yeah, I agree with you.
On the wouldn't give it to a dog comment, I think we should thank Adam for a very engaging and enlightening presentation. For everybody who now knows who Adam is and has an interest in AMR, from soup to nuts, so whether it's molecular biology or the social science behind people taking antibiotics, make your way to his door and see if you can come with some collaborative projects going forward. So we just thank Adam again.
Thank you.